Okay, so next thing. Humans can do glycolysis faster than human lungs can take in oxygen. Yeah, so, and this truth gives me an excuse to show Ronald McDonald doing the bench press, yeah? Why am I showing Ronald McDonald doing the bench press? Well, so he's in over his head, right? His muscles aren't very big, yeah? This is not me. My muscles are much bigger than that. Nevertheless, there's going to be an upper limit to the number he does, yeah? This is kind of like doing push-ups only fancy, yeah? Whether you've done bench press or you've done push-ups, you know what it's like to do those kinds of exercise. How do your arms feel? They start to feel sore. They start to give you a burning sensation. They start to fail, yeah? You can only do so many push-ups, yeah? You can only do so many reps of the bench press, yeah? Does that make sense? And why is that? Well, because what's going on in your muscles is your muscles are doing glycolysis to generate ATP. They're also doing the Krebs cycle and electron transport to generate ATP. But if they don't have any oxygen, then what happens? What happens to the electron transport chain if there's no oxygen? It stops, yeah. The simplest explanation for what happens is the electron transport chain stops. Now, why that happens is because electrons are given to oxygen, right? If there's no oxygen, this guy can't give his electrons to oxygen. And if he can't give his electrons to oxygen, then he can't receive any more electrons from the next guy. And if he can't receive any more from the next guy, then this guy has no one to give his electrons to, and then he can't receive any more from this guy. Does that make sense? You end up with effectively an electron traffic jam. Does that make sense? Is that analogy useful? Okay, so with no oxygen, the electron transport chain stops, and as it turns out, the Krebs cycle stops, right? None of that can happen without oxygen, right? And so we're using a lot of glucose to not get a lot of ATP, right? If we're only getting 2 ATPs per glucose by doing glycolysis, the amount of energy we're getting out of our glucose has been drastically reduced. Does that make sense? Right? We're using glucose a lot less efficiently. All right, moving on. Furthermore, again, as we said earlier, obligate anaerobes can't possibly be doing aerobic respiration. They can't possibly have an electron transport chain, yeah? How could they have an electron transport chain delivering oxygen if they killed in the presence of oxygen, yeah? Does that make sense? All right, so what do we do if there's no oxygen? Well, answer, it depends on what kind of creature you are, yeah? If you're a person, what is building up in your muscles as you exercise what's called supramaximally? You're getting lactic acid in your muscles. How many people know about lactic acid buildup in your muscles? If you've ever exercised, you feel that burn either from doing push-ups or from riding your bicycle up the hill or whatever. That burn, at least up until recently, was widely agreed to be caused by the accumulation of lactic acid. Yeah? What about this picture here? Are we looking at human beings in these casks? What's in these casks? Wine or beer, yeah? Hopefully not people. They'd be dead, yeah? Jimmy Hoffa is in here, yeah? Just a little joke, sorry. Do you guys know who Jimmy Hoffa is? So Jimmy Hoffa is a famous, was a famous union, he was the Teamsters, the truckers union, back when you know, unions used to have strikes and so on. He was the leader of that union, and he disappeared, right? Possibly because he was hanging out with mobsters, or for whatever reason, he disappeared, and so he's sort of become a running joke of what's happened to his body, because nobody ever found his body. So there's speculation that his body was buried in the foundation of what's that? What's the stadium? No, it's where the New York Football Giants play. The Meadowlands in New Jersey, where the Jets and Giants play. People say that Jimmy Hoffa's body was buried in the cement of the foundations of that football stadium. The Simpsons feature a cartoon in which there's a guy running for a long pass, and he ends up tripping over this body-shaped mound in the football field. Anyway, so Jimmy Hoffa's body is not in one of these barrels, hopefully, yeah? So now you know who Jimmy Hoffa is, yeah? Don't say you didn't learn anything in my class today. Okay. So what's in here? So what living, let me ask, it could be beer or wine. What living thing is in there making the beer or wine? Yeast, yeah? Saccharomyces cerevisiae is the microbe that makes, is he making lactic acid when there's a cork in these barrels and there's no oxygen? We do not drink beer or wine for the lactic acid. What do we drink it for? The taste. No, sorry. Sorry, everyone laughs. So there's alcohol in there, yeah? So these guys are metabolically different from people. When people exercise without oxygen, they make lactic acid, yeah? When yeast eat without oxygen, they make alcohol. And that makes our booze, whether it's beer or wine. Does that make sense? You guys got that? Okay, so alternatives to aerobic respiration. There's a couple. There's anaerobic respiration. That's where we use an electron transport chain, but instead of giving the electrons to oxygen, we give them to something else. Yeah? You guys got that? Then there's fermentation, where we don't do anything with our electron transport chain. We just do glycolysis. And as it turns out, both humans and yeast are doing fermentation when we don't have, we're doing lactic acid fermentation and they're doing alcohol fermentation, okay? All right, so before that, anaerobic respiration is where we have different kinds of electron acceptors besides oxygen at the end. Nitrate, carbonate, sulfate, those can accept electrons and get reduced in other things besides humans. So other microbes can do other kinds of anaerobic respiration without oxygen. We don't. We do fermentation, which is where we don't get to use our electron transport chains at all. And instead, we're only generating ATP from glycolysis and making lactic acid as a waste product. So, fermentation, respiration is here. We have glycolysis, Krebs cycle, and then the electron transport chain. Do you see the diagrams of each of those? Yeah? Here we have glycolysis, and that's it, right? We run into a dead end here with various fermentation end products. And the end products can be diverse. There's alcohol fermentation, makes ethanol. And you guys know what ethanol is. This is booze, right? This is the active ingredient in wine and beer. Lactic acid is what we do. You guys following along here? Okay. So without oxygen, yeast make alcohol, and CO2 is a waste product. This is alcohol fermentation. Yeah? So here's our 6-carbon glucose, and I don't know what happened to it. Looks like I got some digital damage to the image of some kind. We make 2 3-carbon pyruvates, and then those get turned into alcohol. Yeah? Humans make lactic acid instead of alcohol. That's where we break apart the glucose and we make a different molecule called lactate. which of these molecules is nastier and more toxic? Lactic acid or alcohol? Alcohol is more toxic. Why do we do lactic acid fermentation? Because it's less. Imagine what we would do if we did alcohol fermentation. What would happen to you when you went onto the exercise bike? Yeah, you would get drunker and drunker, yeah? Right? That would be a, I don't know, you might think that was a good thing, but imagine what would happen in the Tour de France. Whoa, it'd be a bit, sorry. Anyway, what a weird idea, yeah? All right. So, and the reason that yeast make alcohol is because they don't worry about polluting their environment, yeah? Their environment is the environment. Our cells, if they pollute their environment, who are they polluting? They're polluting you, right? Does that make sense? Anyway, so lactic acid creates a very unpleasant burning sensation, makes you suffer, yeah? So many people who exercise work on their lactate threshold and try to make sure that they can work aerobically better. So we've talked about two kinds of fermentation, alcohol fermentation and lactic acid fermentation. As it turns out, their bacteria can do lots of kinds of fermentation, yeah? Here's a couple of kinds. Lactic acid fermentation can be done by lactobacillus, yeah? By strep. Propionibacterium can do propionic acid fermentation. Clostridia can do butyric acid fermentation. There's succinic acid, acetic acid fermentation done by some of these other microbes. And so all of these different products make the complex flavors of fermented foods. Can you think of some foods besides booze, wine, and beer that are fermented? Yogurt is fermented. And when you eat yogurt, how does it taste compared to regular milk? It tastes more sour, yeah? And what do we have? We have lactic acid from lactobacillus bacteria, right? So the sourness, tartness of yogurt comes from the lactic acid waste product that those bacteria make when they're doing fermentation. Does that make sense? Right? When we eat yogurt, we don't get drunk because they don't do alcohol fermentation. They do lactic acid fermentation. Yeah? All right. What else besides yogurt and pickles? Yeah. And pickles have tart and complex taste from the microbes that are fermenting them. Right? How about kimchi? Does anyone eat kimchi or sauerkraut? Yeah? So sauerkraut is German kimchi, or I guess kimchi is German sauerkraut, or is Korean sauerkraut, excuse me. Does that make sense? What's that? What about moonshine? Oh, kombucha, excuse me. I'm deaf. People hear what they want to hear. So kombucha is, I think, a co-fermentation of both yeast and bacteria, yeah? There are others. I always think of the complex, funky smells of cheeses, right? Cheeses are also fermented, and the different tastes of different kinds of cheeses and the different kinds of fermentation create different kinds of flavors. The complexity of flavors of fermented foods comes from the complexity of fermentation products that those microbes can make when they ferment those foods, yeah? Okay. Why do we care? Well, because the way microbes ferment things can help us identify them, right? We can do diagnostics based on fermentation of various products, yeah? You guys with me here? Right? Here's a bunch of and again, you don't need to know all these different kinds of fermentation, but you should know that fermentation is diverse, yeah? Here's one that might be of interest. Have you heard of malolactic fermentation? Have you anyone like to drink Chardonnay? Ooh, lovely Chardonnay. Raise your hand if you like to drink Chardonnay. Raise your hand if you've had people describe a Chardonnay as buttery. Have you raised your? Is that familiar? So some Chardonnays taste kind of sour, and other Chardonnays taste kind of softer and a little bit oily, a little bit buttery. Yeah, and that is because you can give bacteria. This is called malolactic fermentation, and it's done by different bacteria, Enococcus ani and Lactococcus lacti. These are two different guys that can take malic acid. Have you heard of malic acid? Anyone ever eat sour gummy worms? Raise your hand if you like sour gummy worms or sour candy. Yeah? So sour candy, one of the things that they put into sour candy to make it taste sour is malic acid. Sometimes they put tartaric acid in there also, but it's a really sour fermentation product. It's from yeast fermenting grape juice, yeah? But if you can get bacteria, these bacteria in there, they'll take the very sour malate and turn it into less sour lactate. And when they do, the wine gets less tart. It gets softer tasting, yeah? So if you've had a buttery Chardonnay, that is a sort of a very skillful fermentation where you first let yeast ferment to make alcohol, and then once the yeast are done, you get these bacteria to make lactic acid from malic acid. Yeah? Does that make sense? Okay. All right. So, other catabolic pathways. So, can we break down other things besides carbohydrates? Can we get energy from lipids? Sure. So, here are lipids, fatty acids. What happens to them? Well, we break them down, and where do they end up? Looks like they end up in the Krebs cycle, yeah? And then we can predict where they go from there, yeah? What about amino acids? Looks like amino acids, again, we can use amino acids to find out the metabolic characteristics of various kinds of bacteria. We can look at whether bacteria can desulfurylate certain amino acids. Some amino acids have sulfur in them, and Salmonella can desulfurylate the amino acid cysteine, and E. coli can't. So can you see this black stripe here? This comes from Salmonella. So this is Salmonella, and this is E. coli. Does it matter if we know the difference between Salmonella and E. coli? Yeah, Salmonella is food poisoning, yeah? E. coli is something we expect sort of by convention. If somebody has E. coli, generally no big deal, although there are pathogenic strains of E. coli. If somebody has Salmonella, we know they've got dysentery, yeah? Okay, so we can use several metabolic pathways to know more and more about a bacteria until we've identified the species, yeah? We can use a urease test and a nitrate reductase test and other tests in combination with a gram stain or something in order to identify what bacteria we've got. Does that make sense? Okay, and then we do that all day long, and then we can get a decent wage, yeah? And again, we can help patients without having to listen to them or have them yell at you or whatever it is that's making you want to kill your patients, right? If you find yourself wishing you could kill your patient, even though your job is to save them, and if you want to kill them every day, then maybe this is the job for you, yeah? And if you only want to kill them once a month, maybe you're headed for the right career. Does that make sense? Is this touching a nerve for some people? Is this striking a chord for people who actually care for cranky, tyrannical, bossy patients. If it is, again, you have to be nice to those patients no matter how rotten they are to you. It can be very difficult, yeah? So maybe if you're finding yourself having a ball in lab saying, gosh, I wish I could do these little chemical tests all day long. Well, it looks like if you did and you lived in San Francisco, you would start at 60K, yeah? Which is decent, which is more than I make. So, so it's decent moolah, yeah? Guys getting tired? Yes, was that the dumbest question I asked all day?